Good morning, good afternoon, good evening. I'm Professor Jon Frampton, the Professor of Stem Cell Biology at the University of Birmingham.
I'm Dr. Mary Clark, Senior Stem Cell Researcher, also at the University of Birmingham.
I'd like to introduce to you the program, the Master's Program in Stem Cell Biology and Application. So to start, stem cell biology or stem cell science, it's a very large and diverse subject that has a lot of implications for human health, but also much more widely throughout the economy. You're probably aware of all the interest that there is in stem cell science. There's been a lot of expectation and debate around the potential for regenerative medicine for improving health, particularly in older age. But the potential of stem cells goes way beyond that into all sorts of spheres of life. To get a full appreciation of what stem cell science actually is and what it requires, then you have to have an understanding of a broad range of subjects: cell and molecular biology, tissue structure, physiology, practicalities of tissue engineering and bioprocessing, but also areas such as how this gets implemented in the clinic, or in agriculture, or in horticulture. And on top of that, you have all sorts of considerations around ethics and regulation. The program, the Master's Program, which runs through a year, should provide a comprehensive understanding of stem cell biology. And through it will be highlighted key areas of research, and it will encompass all multicellular organisms. The emphasis, or a big emphasis, will be placed on the technologies that are used in stem cell science, such as omics, bioinformatics, and gene editing, about which I'll come back to in a moment. There will be critical evaluation of existing or potential stem cell applications. That will very much encourage amongst the students, because a lot has been said about applications that are current or that may be possible in the future. But these need a proper critical consideration, but also placing them in an ethical and socio-economic setting. On the left-hand side, let me just get a pointer for you, because that will help. On the left-hand side over here is just a list that you shouldn't read at this point, but a list of the 6 compulsory modules. The ones in green run in the first semester, the ones in yellow run in the second semester. In the first semester modules, concurrently, we'll be presenting the first 2 modules. And the aim of these modules is to give you a strong basis in stem cell biology and all aspects that make a stem cell different from other stem cells, other cells. And in parallel with that, the technologies will be described in great detail around bioinformatics, things like core omics methodology, but techniques like flow cytometry and cell sorting and imaging. Following that, still within the first semester, will be a module that talks about pluripotent stem cells, the stem cells that have probably been most talked about in the press, in the media, and in science for their potential in regenerative medicine. In the second semester, again, the 2 modules will be run in parallel because they have certain synergies and crossover. So that will be what we call natural regeneration. That's the body's own processes for regeneration. And at the same time, a module that describes adult stem cells. So all of the tissues in our bodies have stem cells that help them to be maintained throughout life. And we'll cover a variety of these by way of example, but also how it is that those cells might be usefully manipulated within the body, but also how they underlie critical health-related issues such as cancer and the aging process. Should just point out that in terms of natural regeneration, the biology and our understanding of those mechanisms involving stem cells are really very exciting. The example we like to quote is limb regeneration. Now, you may not know, but some amphibians, if they lose a leg, then they can grow a new leg back. But some very related amphibians, so salamanders can do it, as it were, but some related amphibians like frogs can't. But through understanding the mechanisms in amphibians, very recently, literally within the last few weeks, it has now been shown that the things can be tweaked, as it were, in a frog so that it is now able to regenerate its leg. The implications of that, I think, are pretty obvious, that it's now the next step is, can this be done in mammals, and can it be done, of course, in humans? So exciting stuff. Thinking about that regeneration and how stem cells can be employed in replacing tissues, replacing limb parts, etc., then the final module in that second semester will be very much about how all those component parts derived from stem cells, how they get put together, what the processes are, what else is required, and how do you actually do this in a regulated environment, both in commercial terms, but also in clinical terms. The third part, as it were, of the master's program is the dissertation project, which we regard as a very important part. That actually runs across both semesters, and I'll say a little bit more about that in the next slide. So it operates throughout the year. Students will be engaged very closely in this research project. It'll be an independent, novel research project for each student, but there'll be a common theme about those projects in that they will all involve manipulation of cells at the stage of them being a pluripotent stem cell, so like an embryonic stem cell. And the way the technology that will be used will be so-called CRISPR-Cas9 gene editing. This is something which has evolved over the last decade or so, and really it's a quantum leap in our ability to be able to manipulate cells in a both experimental and a therapeutic way. What it means is that we can very precisely change the coding sequence of genes. Now, from a clinical point of view, just by way of example, this means that it would be feasible to change an inherited mutation that has a health effect, and that would be a lifelong change. So the example we like always to give is a disease which you may well be familiar with, which is called sickle cell anemia, which is caused by one base pair change inherited from the individual's parents. That base pair change can be manipulated by gene editing in blood stem cells. It's a disease that affects red cells derived from blood stem cells. That can easily be done using this technology, and that person then, with those stem cells transplanted back into them, would be fully cured for that disease. Now, the aim of the project is that the student, with guidance, will choose an area that is of interest to them in either terms of a particular disease or a particular aspect of biology. And they will be guided as to how they can design a project that enables them to manipulate that gene in these pluripotent stem cells. Having done that, to then see whether the predicted consequence can be reflected in the cells that you can then derive from those pluripotent stem cells by way of differentiation. The teaching approaches are varied. This is a master's level program, so we expect students to work to some extent independently. There will be lectures, but it's not lecture dominated. We will have frequent tutorials where there's a closer interaction between students and amongst groups of students. But particularly, we will encourage student-led seminars and debates. And there will be opportunities for demonstrations and practicals. So thinking, for instance, of the technology module, the methods module 2, then there will be demonstrations there of various aspects of that, for instance, flow cytometry and cell sorting. There will be a lot of self-directed learning. And of course, as I already said in the previous slide, there is an independent laboratory-based research project. So it's a real mixed approach to how you learn during the year of the program. Assessment, again, is also varied, and the flavor of it changes from semester 1 to semester 2. So there will be some, let's say, more traditional exams, short answer questions and very short essays. These are now all done as open book exams, so not sitting in a hall having an examination, but doing it online. That will be predominantly at the end of the first semester in January. As things move forward, but also including in the first semester, there will be in-course problem-solving exercises that will be assessed and essay assignments. And as the confidence and ability of a student develops, then they will be given the opportunity to make scientific posters and oral presentations, and also stimulate debates. A lot of stem cell science has been somewhat controversial, and there are very many areas that would benefit from such debates. There are group projects, for instance, in the bioinformatics part of the program. And again, the independent research project and a written dissertation is a key component. That written dissertation will be in the form of a scientific journal paper, so a real piece of science. As I said, the study of stem cell science is a very broad subject, and the specific skills that will be gained during the year of study are to some extent obvious, but actually more than that and very broad. So it encompasses research methods, cell biology, genetics, omics, bioinformatics, as I've already said. There is a big emphasis on how data is analyzed and presented in the form of research findings, hence the dissertation, but also other aspects of the assessment process. There will be an understanding gained about how you work in a clinical environment, so what sort of research is done that is clinically oriented and how trials are conducted, but also moving towards the practicalities of commercial translation, especially in that last module on bioengineering and tissue engineering. But also the regulatory issues that are part and parcel of that. So the intellectual property, how you patent new ideas, and how you get them approved by health regulatory bodies. And also there's the potential or skills are developed about how it is that a subject such as this deals with the ethical and the social and the political issues that arise. There's also a whole bunch of general transferable skills that will be gained throughout the course. Oral and written communication, that's probably self-evident. Leadership and teamwork, a lot of things will be done in groups. Critical thinking, that's a very important part of the process of learning in this program. But there's aspects like time management, enterprise skills, the awareness of business, and so on and so on. So it's a broad range of both specific and general transferable skills. So what does this set you up for in terms of your career? Where are you going to be going the year after you finish this? Well, you could go into research. You could go into fundamental stem cell biology research. You could go into translational research, which is more geared towards either the clinic or other applications of stem cells in both horticulture and agriculture. So you could be working in a medical or a bioeconomic commercial context as well. So it doesn't have to be necessarily just in an academic research laboratory. You could, with the knowledge that you've gained, actually specialize in an area that does not involve being a researcher or delivering that research in a commercial context. You could be involved in legal aspects of the topic, regulatory areas. You could get into scientific publishing or journalism. I mean, really, there is a vast wealth of opportunities that are opened up to you. So I hope we've given you just a flavor of what's involved, the excitement of stem cell science, and that it's a deep and broad subject. And of course, we will be happy to address any questions should you have them. Our contact email details are there at the bottom of this slide, so please feel free to do that. And lastly, just to say, this image here is a picture of Birmingham, of the university, and I think you can appreciate that it's a wonderful scene. It's very green, it's very historic. So those are the main university buildings there in the center. lovely sports facilities, and I will use my pointer now. Finally, over here is the biomedical environment. So this is where we are, where we do our research on stem cells, and the hospital where a lot of those new ideas are trialed and implemented. So I hope you feel that you would benefit from following our program, and look forward to questions from you. Thank you.